Most people look at their cat's green eyes and see something beautiful, a color, a mood, a quiet kind of mystery staring back at them from across the room. But scientists who spent years mapping the feline genome looked at those same eyes and saw something far more complicated. Because when researchers began tracing the genetic origin of green eye pigmentation in domestic cats, they did not find a clean, simple, and that predates the domestic cat by millions of years. They found a color that, by strict evolutionary logic, should not be as common as it is. And when they traced it back to its source, the trail did not lead where anyone expected. It led to desert wildcats roaming north, Africa 10,000 years ago, to a single domestication event so narrow it still echoes in every cat alive today, and to a pigment cyst. So strange that the same gene controlling eye color in your cat also controls coat color, hearing ability, and immune function simultaneously. Green eyes are not just a color. They are a biological record of one of the most consequential genetic events in the history of the animal kingdom. And the story of where they actually come from has only recently, through advances in feline genomics, been told with any precision at all. The place to begin is with pigment itself, because green eyes are not produced by green pigment. That single fact surprises most people, and it matters enormously to understanding what the DNA eventually revealed. Cat eyes, like human eyes, Haas and Kahn, contain only two types of melanin pigment: eumelanin, which is dark brown to black, and pheomelanin, which runs from yellow to reddish orange. Green does not appear anywhere on that list. What produces the appearance of green is a combination of a low concentration of yellow, why the sky appears blue. Light enters the iris, the short wavelengths scatter, and the interaction between that scattered light and the underlying warm pigment produces the perception of green. The eye is not generating a color, it is performing a trick of physics using whatever pigment concentration genetics happen to assign. This matters because it means green eyes represent a precise calibration. Much pigment, and the iris reads as gold or amber. Too little, and it trends toward a pale gray-green or blue-green. The vivid, saturated greens seen in certain breeds occupy a narrow genetic window, and reaching that window requires a very specific interaction between multi-P. illele gene variants working in concert. A study published in PLOS Genetics in 2020 identified the primary genetic architecture behind feline eye color, locating a key regulatory region near a gene called KITLG, the KIT-LEGAN gene, as a major driver of the blue to gold pigmentation spectrum in cats. Variants in this region, along with secondary modifiers, control how much and what type of melanin gets deposit D in the iris stroma during development. Green eyes, sitting in the middle of that spectrum, are the product of a specific allele combinati. That limits eumelanin without eliminating pheomelanin entirely. Getting there requires the right ancestral starting point, which is where the origin story begins to get genuinely strange. Because the domestic cat, every single domestic cat alive on Earth today, regard less of breed, coat, or eye color, traces its ancestry back to a single subspecies, Felis sylvestris libica, the African wildcat. Riccia RCH published in Science in 2007, led by a team from the National Cancer Institute, and drawing on mitochondrial DNA from wildcat popula. Tie-ins across 4 continents established this with considerable certainty. The domestication of the cat did not happen in multiple places. The ancient Near East, somewhere between 10,000 and 12,000 years ago. The African wildcat that walked out of that desert and into human settlements carried a specific ancestral genome. Every coat pattern, every temperament trait, every eye color variation seen in the 60 million domestic cats alive today is a derivative of that single founding population. But here is what scientists did not initially expect. The African wildcat, the ancestor of every green-eyed cat ever born, does not typically have green eyes. Wild libica populations are predominantly amber to yellow-eyed. The green eye phenotype, particularly the deep, saturated green associated with breeds like the Russian blue or the Egyptian Mao, appears to have emerged after domestication, shaped by a combination of genetic drift in small founding populations, centuries of selective human breeding, and the cascading effects of the domestication syndrome, the cluster of genetic and physiological changes that accompany the shift from wild to domestic life across nearly every species that has undergone it. The green eye, in other words, is not a preserved wildcat trait. It is something that emerged through the process of becoming domestic, and understanding how and why it emerged requires going deeper into the genome than eye color alone. The domestication syndrome is one of the most fascinating concepts in modern evolutionary biology, and it applies with striking force to the domestic cat. First formally described in the context of foxes by Soviet geneticist Dmitry Belyaev through his famous silver fox experiment beginning in 1959, The domestication syndrome refers to a cluster of traits that appear together across species when they are selected for tolerance of humans: floppy ears, reduced skull size, shortened snouts, piebald coat patterns, juvenile behavioral retention into adulthood, and crucially, changes in pigmentation, including eye color. Belyaev's foxes, selected purely for tameness over dozens of generations, spontaneously began producing individuals with spotted coats, blue eyes, and other pigmentation changes that no one had deliberately selected for. The implication was profound. Selecting for behavioral tameness was simultaneously, through some shared genetic mechanism, altering pigmentation pathways. The two traits were biologically linked in ways that took decades to begin untangling. The genetic explanation centers on neural crest cells. These are a population of stem-like cells that migrate throughout the embryo during early development and give rise to an extraordinary range of structures, including the peripheral nervous system, facial cartilage and bone, the adrenal glands, and the melanocytes, the pigment-producing cells responsible for coloration in skin, coat, and eyes. The leading hypothesis, developed by researchers including Adam Wilkins, Richard Wrangham, and Tecumseh Fitch in a landmark 2014 paper in Genetics, proposes that selection for tameness works by selecting for mild neural crest cell deficits. Animals with slightly reduced neural crest cell activity are less reactive, less fearful, less aggressive toward humans, but because those same cells drive pigmentation, mild reductions in their activity simultaneously reduce melanin production across multiple tissues. In the iris, that reduction pushes pigmentation toward the lower end of the concentration spectrum, toward green, toward blue, away from the deep amber of the unmodified wild ancestor. This does not mean every green-eyed cat is tamer than every amber-eyed cat. The relationship between neural crest activity and specific behavioral or pigmentation outcomes is probabilistic, not deterministic, and it operates across populations over generations, not in individual animals. But at the population level, the signature is visible. A study from the Lyons Feline Genetics Laboratory at the University of Missouri, one of the leading centers for cat genomic research globally, found measurable correlations between pigmentation gene variants and the broader cluster of domestication-associated traits when comparing wildcat and domestic cat genomes. The domestic cat genome, compared to its wildcat ancestor, shows evidence of positive selection in genes associated with memory, fear conditioning, and reward-driven learning, precisely the neural pathways you would expect to change if tameness were being selected for over millennia. The pigmentation changes, including the emergence of green and blue eye phenotypes, appear to be a passenger in that selection process rather than its destination. But the story does not end with domestication, because once the domestic cat entered human society, humans began doing something that evolution alone would never have done, deliberately selecting for appearance. The emergence of formal cat breeding as a structured practice in the 19th century, beginning in Britain and rapidly spreading across Europe and North America, introduced a new selective force into feline genetics that operated with a speed and intentionality that natural selection cannot match. Breed standards were written, shows were organized, cats were evaluated and paired based on physical criteria, including coat texture, body conformation, and, crucially, eye color. The Cat Fanciers Association, founded in 1906, and similar organizations in Europe, began codifying what a correct eye color looked like for each breed. For the Russian blue, the standard required a vivid, bright green. For the Egyptian Mao, green was similarly mandated. For the Abyssinian, a rich gold to green. These were not passive preferences. They were selection pressures applied deliberately, generation after generation, narrowing the genetic variation within each breed while pushing specific traits toward extremes that no wildcat population had ever expressed. A 2019 genomic study published in Current Biology involving whole genome sequencing of domestic cat breeds alongside wildcat populations found clear evidence of this artificial selection at pigmentation-associated loci. The variants associated with vivid green eye expression showed elevated frequencies in breeds where green eyes are a standard requirement compared to random-bred cat populations. And those frequencies were far higher than anything observed in wild Libica populations. Human aesthetic preference had, over roughly 150 years of formal breeding, driven a pigmentation phenotype to fixation in specific lineages. The green eye, in its most saturated and distinctive form, is partly a product of nature and partly a product of human taste operating through genetics. It is, in the most literal possible sense, a co-created trait. And what makes this even more layered is the relationship between eye color and coat color in cats, because they are not independent variables. The gene variants that shift eye pigmentation also interact with coat pigmentation pathways through shared melanocyte biology. A white cat with blue eyes carries a high probability of deafness in one or both ears, a well-documented consequence of the W gene's effect on melanocytes in the cochlea, where those cells play a structural rather than pigmentary role. Orange coat coloration in cats is controlled by a locus on the X chromosome, and cats carrying certain orange-associated allele combinations show shifted distributions of eye color phenotypes, compared to non-orange individuals. The biology of color in cats is not a collection of independent switches. It is an interconnected system where pulling one lever moves several others simultaneously. The question that genomic researchers kept returning to, even after the domestication pathway and the breeding selection pressure had been mapped with reasonable confidence, was a deceptively simple one. Why green specifically, not why pigmentation changed during domestication? That part the neural crest hypothesis addresses convincingly. But why, across so many independent cat populations on multiple continents, across breeds developed in Russia and Egypt and Britain and Southeast Asia with almost no shared breeding history, did the intermediate pigmentation phenotype so consistently resolve as green rather than some other color? The physics of Rayleigh scattering produces blue at very low melanin concentrations. Amber and gold dominate at higher concentrations. Green sits in the middle, and that middle position turns out to be where a surprising number of domestic cat populations cluster when left to breed without intensive human color selection. Understanding why requires looking not just at the pigmentation genes themselves, but at the broader population genetics of the domestic cat, and what happened during the geographic spread of the species out of its original domestication center in the ancient Near East. After the initial domestication event roughly 10,000 years ago, cats did not stay in the Fertile Crescent. They spread, riding alongside human agricultural communities as those communities expanded, serving the same function that had made them valuable to begin with, controlling rodent populations in grain stores. They reached Egypt by roughly 4,000 years ago, where the archaeological record shows them transitioning from tolerated pest controllers to genuinely revered animals. They entered Europe through Phoenician and Greek maritime trade, reached India through overland routes, and eventually arrived in East Asia, sub-Saharan Africa, and the Americas through European colonial expansion. Each of those dispersals was a new founder event, a small population breaking off from a larger one and carrying only a fraction of the original genetic diversity with it. And each founder event, by the mathematics of population genetics, increased the probability that intermediate frequency alleles would either fix or be lost entirely in the new population. A comprehensive genomic study published in Nature Ecology and Evolution in 2017, led by Eva Maria Gagel and Thierry Grange at the Institut Jacques Monod in Paris, traced the spread of domestic cats through ancient DNA extracted from cat remains at archaeological sites across Europe, the Middle East, and Africa spanning 6,000 years. The results revealed two major waves of cat dispersal. The first followed the expansion of Near Eastern farming cultures westward into Europe. The second, much later wave, centered on Egyptian cats, spreading across the ancient Mediterranean world through maritime trade networks. Both waves compressed genetic diversity at each step of the journey. The allele frequencies present in the founding population of each new region reflected not the full diversity of the source population, but a statistical sample of it, shaped by whichever individuals happened to make the journey. This repeated bottlenecking had consequences for pigmentation gene architecture in cats, meant that the intermediate green-producing allele combination had a higher probability of being retained across multiple independent founder events than would be expected by chance alone. Researchers at the Feline Foundation and collaborating university laboratories have noted how alleles would be. The exact nature of those pleiotropic effects remains an active area of investigation, but the signal in the population data is consistent enough to suggest that green eye alleles were not merely drifting passively. Something in the biology was helping them persist. That something may relate to immune function. The KITLG gene and its signaling pathway are not exclusively pigmentation machinery. The KIT ligand, also known as stem cell factor, plays critical roles in hematopoiesis, the production of blood cells, in mast cell development and function, and in the regulation of certain immune responses. Variants in this pathway that shift its activity level in ways that produce intermediate iris pigmentation may simultaneously shift immune parameters in ways that have fitness consequences, entirely unrelated to eye color. Research on the relationship between pigmentation pathway genes and immune phenotypes in cats is substantially less developed than comparable research in humans or mice, but the framework is biologically plausible and supported by analogous findings in other species. A 2021 review in Frontiers Tissues creates inevitable functional overlap between genes originally classified as pigmentation genes and genes involved in pathogen response. The implication, still preliminary but scientifically grounded, is that green eyes in cats may not be a purely cosmetic phenomenon with no biological relevance beyond appearance. They may be the visible surface of a deeper genetic configuration that touches immune competence, developmental stability, and the specific set of neural and physiological adjustments that made the domestic cat viable as a human companion species. The color your cat shows you across the room when the light catches their eyes at a certain angle is not arbitrary. It is the outward expression of a system that runs far deeper than pigment. The more researchers sequence the feline genome, the more layers they keep finding beneath that surface. There is one more layer to this story, and it is the one that pulls everything together in a way that researchers did not fully anticipate when the Feline Genome Project began. Because once the genetic architecture of green eye pigmentation had been mapped, once the domestication pathway had been traced, once the founder effect bottlenecks had been identified across the geographic spread of the species, scientists turned to a question that had been sitting quietly at the edge of the research for years. What happens when you look at the green-eyed cat breeds with the most ancient documented histories and sequence their genomes against the wildcat populations closest to the original domestication center? What you find when you do that comparison is not what simple domestication theory would predict. You find that certain green-eyed cat populations, particularly those with documented histories in Egypt and the Eastern Mediterranean, carry a pattern of genetic variation that suggests not one, but potentially two distinct waves of genetic contribution from the wild. Ancestor population, separated by thousands of years. The 2017 ancient DNA study from the Institute Jacques Monod had already established the two-wave dispersal model for domestic cats broadly. But subsequent analysis of pigmentation-associated loci specifically revealed something additional. The intermediate pigmentation alleles associated with green eye expression were disproportionately represented in the second wave, the Egyptian maritime dispersal, compared to the first Near Eastern farming wave. This suggests that the green eye phenotype, or at least the allele combinations that most reliably produce it, may have been concentrated and possibly amplified within the Egyptian cat population during the roughly 2,000 years. The cats spent in Egypt before the maritime dispersal carried them westward across the Mediterranean. Egypt was not simply a waypoint in the geographic spread of the domestic cat. It was a genetic crucible where specific traits were concentrated, possibly through deliberate human selection tied to religious and cultural preferences, before being exported to the rest of the ancient world. The implications of that finding extend into breed history in ways that cat fanciers and geneticists alike are still working through. The Egyptian Mao, widely marketed as the most ancient of all domestic cat breeds, and the direct descendant of the cats venerated in ancient Egypt, was actually reconstructed in the 20th century from street cats in Cairo and Rome by an exiled Russian princess named Natalie Trubetskoy, who brought several individuals to the United States in the 1950s and established the breeding program that produced the modern breed. For decades, the claim that the Egyptian Mao represented direct genetic continuity with ancient Egyptian cats was treated with skepticism by geneticists. But a genomic study published in 2022 using ancient Egyptian cat remains provided partial support for a more nuanced position. While the modern Egyptian Mao is not a pure unbroken lineage from antiquity, it does carry a higher proportion of alleles associated with the second dispersal wave, the Egyptian maritime population, than many other Western breeds. The green eyes, mandated by the breed standard may, in that specific case, represent a genuine if indirect connection to the pigmentation patterns concentrated in ancient Egypt, transmitted not through an unbroken breeding line, but through the geographic source population from which the breed's founders were drawn. The Russian blue presents a parallel case from the opposite end of the continent. Originating in the port city of Arkhangelsk in northwestern Russia, the breed was reportedly carried to Britain by sailors in the 19th century and subsequently developed into a formal breed with vivid green eyes as a defining standard requirement. Genomic analysis of the Russian blue places it within the Western European cat genetic cluster rather than in a distinct Russian lineage, suggesting that the breed's founders were likely drawn from the same broadly Western European domestic cat population that spread through maritime trade centuries earlier, rather than representing a genuinely isolated Russian wildcat-derived lineage. The vivid green eyes that define the breed were almost certainly present in the founding individuals, and then intensified through selective breeding, but their ultimate origin traces back through the same chain of founder events and domestication-related pigmentation shifts that produced green eye expression across the European domestic cat population generally. And what the full genomic picture reveals, when you step back and look at it complete, is a story of remarkable biological coherence underneath what appears on the surface to be arbitrary variation. Your cat's green eyes are not a random accident of genetics. They are the terminus of a chain of events that begins 10,000 years ago in the Near Eastern desert, runs through the neural crest cell biology of domestication, passes through the population bottlenecks of geographic dispersal across 3 continents, gets shaped and concentrated by 2,000 years of Egyptian cultural selection, and arrives finally in the specific allele combination sitting behind. Your cat's iris, bending light into the color that stops you across a room. Every step of that journey left a mark in the genome. Every founder event, every selective pressure, every generation of isolated breeding contributed something to the final configuration. The color is the summary of the history. This is what modern feline genomics has delivered that no previous discipline could. Not just the identification of which genes control which traits, but the ability to read the trajectory of an entire species through the variation present in living individuals. A green-eyed cat sitting on a windowsill in London or Cairo or Tokyo carries inside every cell a molecular record of one of the most consequential relationships in human prehistory, the moment a wild animal decided that proximity to humans was survivable, and the 10,000 years of mutual adaptation that followed. Researchers at institutions including the Broad Institute, the WinFeline Foundation, and the University of Missouri Feline Genetics Laboratory continue expanding the reference genome and deepening the population analysis, with ongoing projects aimed at connecting ancient DNA findings to modern breed diversity at a resolution that was impossible even a decade ago. The green eye looking back at you is not just beautiful, it is a document. It is a 10,000-year record written in pigment and light and the accumulated consequence of every migration, every selection pressure, every narrow passage through which your cat's ancestors passed and survived. Science is still reading it. And the further into the genome researchers go, the more the cat, that quietly sovereign creature sharing your home, turns out to be one of the most genomically interesting domestication stories on the planet. Not because it is the oldest or the most studied, but because the relationship it represents, chosen rather than forced, maintained across 10 millennia through mutual benefit rather than human dominance, left a different kind of mark on the genome than any other domestication event in history. And that mark is still visible every time a green-eyed cat turns to look at you, in the particular way that light moves through an iris shaped by 10,000 years of shared history.